Hello, and welcome to the North Central Region Water Network's first, hopefully annually or on some similar period, virtual harmful algal bloom research symposium. My name is Eugene Bregg. I work for Ohio State University Extension, and I will be the moderator for this afternoon session. The symposium is hosted by the Algal Bloom Action Team, of which I'm a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and University Extension within each state in the North Central region. Today is the second session of the symposium, and here we'll be focusing on preventing and treating harmful algal blooms. It will last 2 hours and feature 3 presentations on emergent harmful algal bloom research, the work of leading researchers across the North Central region. There will be a 10-minute break at 1:45 Central, that's 2:45 Eastern. Following today's presentations, we will host a 1-hour facilitated discussion on the topic of the thematic session to discuss how we can better disseminate information regarding harmful algal blooms. This discussion will help facilitate the work of our action team, of course. Our team is working to develop a website of resources, including fact sheets and FAQ pages on harmful algal bloom topics. Additionally, we plan on organizing webinars, recording video content, continuing to host this seminar into future years as well. However, to do that, we are seeking your insight and input on how we can improve dissemination materials and content areas of research for the public. Because of the number of people at the symposium and platform constraints, an additional sign-up was required for the facilitated discussion, and all the spots have been filled. We apologize for the inconvenience if you were not able to sign up in time. We have been blown away by the interest in this event, and we'll make sure we can accommodate additional folks in future events. If you have questions about the facilitated discussion, or if you are having any technical issues, please note that the chat box and our team will do their best to assist you. And of course, if you have questions for our presenters, please place them in the question and answer panel, and I will ask them of our presenters to follow as time allows. And in case you're not familiar with the platform, hover around on the Zoom window and you should see a little question and answer button appear. Before we get underway, I would like to take a moment to thank our sponsors for making the symposium possible. In addition to the support of the North Central Region Water Network and the National Network of Water Research Institutes, we want to thank especially our champion sponsors, FlowCam by Yokogawa Fluid Imaging Technologies and Limnotech for their support. Thank you, sponsors. So let's get started. The topic at hand is preventing and treating harmful algal blooms, and we have 3 fantastic speakers from the region lined up for you today, and some a little bit outside of the region, but close enough. We will begin with John Lenhart, a professor of environmental engineering and co-director of the Water Resources Center at The Ohio State University. He received his PhD in environmental science and engineering from the Colorado School of Mines in 1997, and he currently investigates contaminant interactions with interfaces in heterogeneous systems with an emphasis on the role of natural organic matter. You can read the rest of the bio at your leisure. I will also add that I came to my role with the Algal Bloom Action Team through a small grant that John and I shared a responsibility for in Ohio. And I'm very glad that John could give us some time to present today. Thank you, John. I'm going to stop sharing my screen so you can share yours.
Thank you for that introduction, Eugene, and good afternoon, everybody. Wow, the participation that I see is pretty impressive, over 350 people. So I'm going to try to set up my share, and I believe I have it successfully shared now. Thank you. All right, so as Eugene indicated, today I'll be talking a bit about removing of cyanotoxins during the treatment of municipal drinking water. I'd like to first off acknowledge that this work is been done by students in my group, such as Yujo, Yenling, Kate, and Eshnika. So I want to acknowledge their efforts. And Also acknowledge the sponsorship for this research. Much of it came through Ohio Department of Higher Education through their harmful algal bloom research initiative. So I'll talk a little bit about that, as well as the Ohio Water Development Authority. And so in addition to my role as a faculty member in the Department of Civil Environmental Engineering at Ohio State, Eugene also indicated that I'm the co-director for the Ohio Water Resources Center. So to begin, what I would like to do is, get to the right screen. Okay, so I think everybody recognizes in 2014 that Toledo had some issues with their drinking water. So one consequence of that was the agencies and university researchers in the state of Ohio got together and organized to develop this cooperative research program, which is called the Harmful Algal Bloom Research Initiative. And this funding for this initiative was provided through the state of Ohio by the Ohio Department of Higher Education. And the whole program is administered through Ohio Sea Grant. And then what it does is fund sponsored research throughout the state from other research universities. So this is a nice cooperative interaction between researchers and agencies, and it's done a very good job with respect to trying to identify and deal with some of the issues in Ohio that are certainly important for other states in the region. So this particular program has, in terms of its priorities, the four shown here. So looking at issues with respect to bloom formation, methods to detect toxins, drinking water treatment, and enhancing engagement. So my specific role within this overall initiative has been to look at methods to treat drinking water. And at the beginning of this initiative, what they were looking for were specific approaches that utilities could use to enhance their existing treatment in order to deal with the toxins that are being produced during these harmful algal blooms. So much of what we did was looking at the use of activated carbon. So that's pretty much what I'm going to talk about today. So when we're looking at, in general, toxin removal, we have sort of three broad characteristics that we need to deal with. We need to understand characteristics of the toxin, characteristics of the water we are trying to treat, and then characteristics of the system itself that we're looking at. So points of importance with respect to the toxin are the form, how much of the toxin is found within the cells versus within the water as a whole, because that makes a big difference in terms of treatment. Concentration of the toxins that we're interested in. So this is obviously going to vary. And in cases when intercellular toxins are important, usually be a much higher concentration within those cells themselves. And then the specific types of the toxins are also important to understand, because whether it's a microcystin or saxitoxin or different variants of microcystins or saxitoxins, then their properties vary, and that's important to understand. And then the water characteristics itself need to be understood. For example, the pH is important because it impacts oxidation processes. And as our research has shown, as well as others, it's important for understanding the removal using activated carbon. So that's what AC refers to. We also need to look at natural organic matter. And natural organic matter is important because it interferes with many treatment methods. And that's going to be a function of the system condition and properties of natural organic matter. And then the ionic strength or salinity is also important because it affects electrostatic interactions, which in some systems are very important. And then treatment-wise, we need to understand which processes are in place for treatment. Is it a conventional, or what I'm calling a conventional plus, that might have the flexibility to utilize additional treatment processes outside those which are considered conventional treatment processes? What about the bloom itself? Is it a seasonal bloom, or is it fortunately a chronic one? In which case, the approach is going to be different. And then resources, does the utility have the financial resources and the human resources, both in terms of manpower and skill, to manage the issue? So these are things that we need to look at. So in terms of approaches, so these are some of the approaches that have been tried. I'll just kind of walk through these real quick. So conventional treatment, so that is shown in the schematic here. So that is comprised of coagulation, flocculation, and then sedimentation to remove the flocculated particles, typically followed by rapid sand filtration as kind of a final polishing step to remove most of the residual particles, and then a disinfection step before it is stored within a clear well or sent to the distribution system. In some cases, dissolved air flotation can be used in place of the coagulation approach. This is particularly well suited for light particles such as those that algae are. So dissolved air flotation is a pretty effective approach for removing algae. In some systems, particularly smaller systems, they would substitute slow sand filtration for the typical rapid sand filtration, and then this is has some advantages in terms of cell removal. And then some utilities have filtration approaches, could be ultrafiltration or nanofiltration, and these have certainly benefits associated with removal of dissolved toxins as a function of the pore size. So ultrafiltration would have a larger pore size, so it allows for a higher flux. But on the other hand, that larger pore size is not particularly effective for removing many of the toxins. The nanofiltration with its smaller pore size is better suited for toxin removal, but on the other hand, you can't treat as much water through nanofiltration type processes. Then the activated carbon. So now we're looking at approaches that might be in addition to these. So this would be either powdered activated carbon or granular activated carbon, so PAC versus GAC. So those have advantages and disadvantages, and depending on their use, and will be a function of whether it's a chronic versus a seasonal and the size of the facility. Ozonation is often utilized and can be used as a pre-ozonation step early in the process or later in the process for removal, and chlorination. So if we look at sort of how effective these different approaches are, so this diagram shows, so these are intercellular, the red circle would be extracellular, and we have our conventional dissolved air flotation, slow sand filtration, ultrafiltration, nano, activated carbon, ozonation, chlorination. Effective to ineffective is sort of the range of performance here. So what we can see is that approaches that exploit physical removal of cells will be good for intracellular toxin removal. So conventional treatment is pretty good for dealing with cells and the intercellular toxins that they may host. Dissolved air flotation is very good at cell removal, as is slow sand filtration, and then our membrane filtration techniques. These, on the other hand, are not particularly well suited for the extracellular, so the dissolved toxins. Slow sand filtration is fairly effective for both because the slower flow rates allows for a more robust biological treatment in these sorts of scenarios. And then over here, we can see these processes are primarily utilized for the dissolved toxins, the extracellular toxins, and are not very well suited for the intercellular toxins. And so in our particular research, we're focused on the extracellular toxins, and we're particularly focused on microcystins. And there are two reasons with respect to why that is. Back when we started this research about, I guess, half a dozen years or so ago, the concern within the state of Ohio coming from the agencies was on microcystins. So that's what we focused on. And the concern was also on the extracellular form of the microcystin. So as we were getting funding through the HABRI initiative, that's what we focused on. So within Ohio, and certainly most other places, the microcystins are going to be one of the more common cyanotoxins. And for utilities that are facing seasonal use, activated carbon, particularly powdered activated carbon, is often the first line of defense. And when we started doing this research, there was a lot of uncertainty over which type of activated carbon to use, how much of it to use, how effective it would be for the different forms of microcystins, the different variants of microcystins. So that's what we were looking at. And so I think what is recognized is the removal is going to depend upon the toxin, the organic matter, water conditions like the pH, and then the actual properties of the PAC. For systems that are facing more chronic issues, they're going to need a more permanent solution, which is often called a multi-barrier solution. So for example, using the granular activated carbon, particularly when it's allowed to develop a biofilm. So that would be the biologically activated carbon, so the BAC. So that works fairly well. Research that we've done, which I'm not going to actually talk about today, shows it works well and is a bit more resilient than is regular granular activated carbon. And then ozone also works well, but one of the issues with respect to ozone is the byproducts that it produces, and those may still have some of the residual toxicity associated with the original microcystin. And then in combination, ozone, a pre-ozonation step with a BAC, sort of couples the benefits of both the granular or biologically activated carbon as well as the ozone, and that's shown to be also fairly resilient in terms of long-term removal. So with our particular research, what we were looking to do is develop guidance that utilities in Ohio could use to help them determine how to best use the activated carbon in their processes, whether that's granular activated carbon or powdered activated carbon. So what I will be talking about specifically is our work with the powdered activated carbon. And what we've done is look at removal as a function of the properties of the carbon. We've looked at different forms of microcystin to try to understand how the chemical and physical characteristics of the microcystin toxins affect removal. We've looked at water composition. So we've looked at different pH conditions, different types of organic matter, different concentrations of organic matter to understand how that affects removal. And then also looked a bit at how operating conditions look at this. So for example, contact time is an important issue. We've also looked at the influence of other water treatment chemicals such as coagulants. That's work that's ongoing, how that actually affects the removal as well. So to begin with, just kind of walk through some of the materials and methods that we utilize. So I imagine most folks are familiar with the microcystin, but in case you aren't, we'll just kind of walk through the structure of the microcystin. So it's a cyclic heptapeptide. The base structure of the microcystin is shown here. So we have the common amino acids, this ADDA group, a glutamic acid, this MDHA group, that group, and then this methyl isoaspartic acid over here. And then we have two variable amino acids that can vary. And obviously, because of those variations in those amino acids, the different microcystin variants will have different properties. So that could be in terms of different pK values, which changes the net charge of the molecule, can change the molecular weight or size of the molecule, and changes hydrophobic characteristics. So there are many different variants that exist, and depending upon which literature or resource you look at, those may be more than 100. I think I've seen some report that there's more than 200 different microcystin variants, so quite a few. But what we looked at were a selection of microcystin variants that Ohio EPA had indicated were most prevalent in the waters that they were monitoring. So those three are shown here. So we've got microcystin RR, the microcystin LR, microcystin LA. So from here on out, I'll call this MCLA, MCLR, and MCRR. And so again, these are differentiated on the basis of the variable amino acid groups that they contain. So if we start with our RRs, that has two arginine groups. So that's shown circled in the blue and the red. And so if we swap out this arginine group with the leucine group, then we get the microcystin LR. And if we swap out this arginine group with the alanine group, then we get the microcystin LA. Consequence of this is we get differences in the properties. So as we change from our microcystin RR to our microcystin LA, we add protonated amino groups to the system, so those counteract the negative carboxylic acid charge, and so we go from they don't counteract, excuse me, so they're neutral, substituting a neutral for a positive, so we go from a 0 to a -2. They're smaller groups, so the molecular weight goes down from 1038 to 910, and it becomes more hydrophobic as we go from the microcystin RR to the microcystin LA, just on the basis of the relative hydrophobicities of these different functional groups. We looked at one more microcystin, and that involves the desmethylated microcystin RR, where this particular methyl group on this methyl isoaspartic acid amino acid within the structure is lost. So this is what we're going to call the DASPRR. So that differentiates it from the microcystin RR. So we have 4 that we were looking at. And in terms of the powdered activated carbon, so we looked at a variety of different powder activated carbons, but converged to these 4 right here. And these were 4 that were commercially available within Ohio, and some of which were being used by different utilities in Ohio. So we have products that have as their source material wood, coconut, a coal blend, and a lignite coal, have different activation methods. We can measure what's called the point of zero charge of these materials, which is the pH at which the surface charge is neutral. If you have a pH greater than that, then the carbon will have a negative charge. If you have a pH less than that, then the carbon will have a charge. So for the systems we were looking at, these carbons, except for this hydrodarco, because we haven't had a chance to measure its pH PCC yet, these 3 at least would all be having a negative charge for the systems that we're looking at. So we've characterized the properties of these. So we measured the surface area. So the wood has a surface area of about 1,500 square meters per gram. And then the WPC is around 600. The water carb is around 800, and then the hydrodarco is around 500. So these are the corresponding iodine numbers that are associated with these. So these are not values that we measured. These are values provided through the vendor. So in many cases, folks just rely on the surface area or the iodine number to try to help them select which product to use. What we would advocate is trying to get more information about the carbon, and what I'll point out is specifically the importance of what's the mesopore volume. So this is a specific subset of the overall pore volume, which is focused on pores of a specific size. So to help us compare results for these different carbons, we cleaned and sieved our carbons to have a uniform particle size from 0.45 to 44 microns. So in terms of our natural organic matter, we looked at 2 different forms of natural organic matter. One is a terrestrial natural organic matter, which is isolated from the Suwannee River. So this is Suwannee River fulvic acid. So this is a product that's available through the International Humic Substances Society. And then the other one that we looked at was one that we isolated ourselves from Grand Lake St. Mary's. So this would be a microbial natural organic matter. So this will allow us then to compare some of the properties of natural organic matter to see how much of an effect that has. One benefit of using the Suwannee River is that since that's available and can be purchased, a variety of researchers could use that, and then the results can be compared to see, it gives a more robust comparison. So our experiments are done in batches. So we would combine our water, and we used a synthetic model water, sort of described here, which mimics the average Ohio freshwater, has a pH anomaly around 8, has alkalinity and hardness that approximates the average Ohio surface water. And again, we did this in order to allow us to compare our results across the different systems that we're looking at, and compare systematically the influence of the carbon or the organic matter or pH in these systems. We looked at different ranges of organic matter as measured by TOC using both our Suwannee River fulvic acid and the Grand Lake St. Mary's fulvic acid. So we combine the water with the microcystin. We form a slurry with our powdered activated carbon, add an aliquot of that, put that in our amber glass bottles, and then shake that for a period of time, collect samples at designated times, filter those to remove the carbon, and analyze the concentration of the microcystin in solution. We've done some analysis using LC-MS, but for the experiments I'm talking about today, we were using just liquid chromatography, and for lower concentrations, ELISA. And so that would get us down to concentrations on the tenths of a microgram per liter. All right, so let's go through some results. So we looked at two different types of experiments. So we start off by looking at what are called kinetic experiments. So here we're looking at the amount of our toxin absorbed per mass of our adsorbent, so milligrams per gram per time. And in this particular plot, we looked at three different NOM concentrations. And so by looking at data over time, it gives us information about both the extent of absorption as well as the rate of absorption. And so these systems, we looked again across a range of carbon concentrations, across a range of toxin concentrations, carbon concentrations, and we did this over a 3-day period. And then we fit different kinetic models to this. And in the end, what's called a pseudo first order kinetic model provided the best fit to our whole data set. And what that does is give us a rate constant, allowing us to understand which carbon absorbed the toxins most rapidly. And it also gives us surface coverage, which allows us to understand which carbon, for the conditions that we looked at, would absorb the largest amount of the microcystin. And then on the basis of our understanding of when we're seeing steady state or equilibrium, we can do our equilibrium systems. And so those are done over a 3-day period. And so this gives us information about the capacity at equilibrium for the different carbons. And from here, what we're looking at is, again, it's the adsorption as a function of the concentration in solution at equilibrium. So again, in this case, what we're just showing is data at three different NOM concentrations. And so on the basis of different isotherm approaches, we can fit an isotherm model to this and get information about partition coefficients, capacity values, depending upon which type of isotherm model that we looked at. So I'm going to start by just kind of quickly reviewing some of our kinetics. So again, we fit our data to a pseudo first order kinetic representation. So that's this expression down here. You can see an example model fit in this diagram here. So this is for microcystin LA. And in this case, this is for the Grand Lake St. Mary's organic matter. So here we're looking at, again, the adsorbed concentration in a mass of toxin per mass of carbon versus time. And the shaded areas represent the 95% confidence interval over our nonlinear fits to the data. And then here we were looking at three different carbon types, so a wood, a coal blend, and a coal based. The summary in this table here is the parameters we would get from our model fits. And so we can see for this particular data set that the capacity for the wood, about 4.6, is greater than the coal blend and the coal base. So that's evident from the data itself. And we can also see, based upon our model fit, the rate constants associated with these. And so we can see for the wood that the rate is quite a bit faster than it is for the coal blend or the coal base. So we're looking at about 3 to 4 times faster for the wood compared to the coal. So in general, that's what we saw across all our data sets. So for the most part, all of our microcystin variants were absorbed to a greater extent and more rapidly to the wood-based carbon than it was to the other. Well, term of this is our coke-based carbon was a fairly poor adsorbent for the microcystins. And that doesn't indicate that coconut is not a good PAC. It's just not a good PAC cystins. And that's something that I'll address here in a little bit. And then what we also saw is that the microcystin RR and the desmethylated microcystin RR, in general, they exhibited more favorable uptake parameters than did the microcystin LR and LA. So they were absorbed more rapidly and to a greater extent. And then for the coal blend and coal based, we didn't see a general specific trend that would allow us to differentiate those two from one another because it's tended to depend upon the system that we were looking at. So that will become more evident here. And what we'll talk then for the remainder two specific time points. So we then are going to look at comparing our results at a 2-hour contact period and then an 8-hour contact period. And so these are useful time points for because they correspond to design criteria that are often utilized. So the 2 hours would be the minimum contact time within a coagulation, flocculation, sedimentation based upon standards that are utilized in the region. And then some of the utilities have the capability to have a longer contact time, so up to 8 hours in some instances. So that would give us an approximation sort of removal they would see at those time points. And what I want to point out is, again, that particularly for 2 hours, what we can see is that 2 hours, if we're looking at this trend and data here for these two carbons, that the results that might be collected from, say, an isotherm under equilibrium conditions would not be a good approximation for what might be seen for a much shorter contact time. So the contact time needs to be understood in terms of making sure that decisions being made are appropriate. So I'll start out by looking at the influence of the carbon type. So what we have now is the percent removal shown here versus our different carbon types. So we've got our wood, our coal blend, and our coal based at our 2 time periods. So here's our 2 hour, here's our 8 hour, and here's our 4 different microcystin types. So the bars then represent the removal for those 4 different microcysteines on these different types of carbon. So we can see, we'll start with the 2 hour, that as was indicated earlier, the wood-based carbon performs the best. So we see removal in 2 hours of, say, roughly 70 to more than 90% of all 4 of our microcystin types. And then under the same conditions, the coal blend and coal based, we see removal that's on the order of, say, 30 or 20 or 30% up to 60%, depending upon the different microcystin type. As we go to an 8-hour contact time, overall removal increases, but the trends observed at 2 hours are still evident. The wood-based carbon performs very well, anywhere from 90 to near 100% removal. The coal blend and coal based are not nearly as good, particularly for the microcystin LA, which is this blue, and the microcystin LR, which is the red. And what we see in general, and this is at 2 hours as well as 8 hours, is, and this is what we've talked about previously, is our microcystin RR and the desmethylated RR. They're removed to a greater extent than are the LR and the LA. So what this reflects is that the properties of these different carbons are different. So the carbons will have a different proportion of pore types. So they'll have macropores and mesopores and micropores. And nominally, those are differentiated on the basis of the macropores having a size that's 50 nanometers or greater. Mesopores can be between 2 and 50, and the micropores are going to be less than 2. So our microcystin has dimensions that range from a little over 1 to around 3 nanometers. So carbons that have a lot of micropores may not be well suited to remove microcystins because of the size of the microcystin. They will not be able to access those micropores. So the carbons that have a lot of mesopores, so those are going to be carbons that are better suited to remove the microcystins because they will be able to access those pore spaces and get to the interior surface area where most of the adsorption sites are. So if we look at how the pore volume is distributed across these carbon types, what we'll see is that the wood and the coal blend, they have similar proportions of the micropores. Coal-based is a little bit less, but what we can see is the wood has much more mesopores than does the coal blend and the coal-based. And it's not necessarily obvious, but the coal blend and the coal-based both have fairly similar mesopore mesoporosities, which is consistent with their overall removal being similar, but they're much less than the wood. And this is particularly evident at these lower contact times. So the next thing we looked at is comparing the microcystin type to see if we can determine more clearly the trends for the different microsystem variants. So the data is pretty similar, but in this case, what we're showing is the percent removal as a function of the different microsystem types. So we have our microsystem LA, LR, RR, and here's our desmethylated RR. And then the colored bars are then our wood, our coal blend, and our coal-based. So as was indicated earlier, and it's pretty evident here, the wood performs the best across all our microsystem variant types. And then what we can also see is the microsystem LA and LR, their removal is lower, and that's pretty consistent across all the carbon types as well, compared to the microsystem RR types. And so we see similar trends as we go up to around 8 hours. And so if we look at the properties again, so we can see the charges are different, the molecular weights are different, and the hydrophobicities are different. So for example, if we look at the molecular weight, we can see from RR to LA, the molecular weight goes down. So on the basis of the molecular weight, we would anticipate that the smaller microsystem LA might be removed to a greater extent. That's not what we observed. So it doesn't seem like the molecular weight is a big determinant in removal. Hydrophobicity, so we would see a similar trend. On the basis of the hydrophobicity, the LA should be removed to a greater extent, but that's not what we see. But what we do see is that there's a dependence on charge. So the microsystem RR is a neutral charge. The microsystem LA is a net charge of -2. And so our overall trend seems to indicate that as the net charge of the microsystem goes from 0 to -2, we see lower removal. And that's a function of electrostatic repulsion between the negatively charged microsystem LA and the negatively charged carbon surface. And if we compare the PAC type to the properties of the microsystem variant, what we see is that the PAC type, again, is a more important factor than is the property of the variant. So what we'll look at next is concentration of natural organic matter. So here we're looking at removal versus concentration of fulvic acid, 0, 5, and 10. So this would give us a TOC of 0, 2.5, and 5. And here we have our different carbon types. And so again, what is evident is our wood performs the best. The coal blend and the coal-based are similar. There's something interesting going on with the coal-based and the microsystem LR. It does not remove the LR very well at all. This is something we're still trying to understand. We don't have a good reason for this as of yet. But as we add organic matter into our system, we see a decrease in removal, less evident for the wood-based than it is for the coal blend and the coal-based. So this is just an indication of the competition with the natural organic matter. What's interesting is that as we increase from, say, our Suwannee River fulvic acid of 5 to 10, is we don't see a big decrease in removal. So it appears there is a threshold above which the concentration of the organic matter does not necessarily increase or decrease removal. This was something we didn't necessarily expect. And then again, if we look at, so again, this is a competition. So our microsystem molecule is competing with our NOM, this little purple blob, to access our pore spaces, our sites within those pore spaces to absorb. And so if we compare, again, our general trends, it does appear that overall the type or the properties of the carbon are a more important factor than are the properties of the microsystem variant, and then is the concentration of natural organic matter. Lastly, what we wanted to look at was how the different types of organic matter, how that influences removal. So we did experiments comparing the removal with our fulvic acid from a terrestrial source, that's the Suwannee River fulvic acid, with the organic matter from the Grand Lake St. Mary's. So Grand Lake St. Mary's, for those of you that aren't familiar with it, is a shallow eutrophic reservoir in Ohio, has chronic bloom issues, and it's a nice system to study issues with algal toxins and harmful algal blooms. see is again removal as a function of microcystin type for our different carbon types. And overall, what we see is there isn't significant difference. We see perhaps slightly better performance of our carbon in the Grand Lake St. Mary's than we did for the Suwannee River fulvic acid. So perhaps for this particular system, the fulvic acid, the terrestrial fulvic acid competes to a greater extent. But one thing I want to note is that previously we had collected some Grand Lake St. Mary's organic matter at a different time of the year, at an earlier date, and we saw some slightly different results. So we're trying to better understand how the organic matter from Grand Lake St. Mary's varies as a function of season, to see if there's some properties of the organic matter that are influencing that. So for these systems at a 5 milligrams per liter of organic matter, their carbon concentrations are pretty much the same, but we would notice that their nitrogen concentrations are quite a bit different. So the microbial is going to have a much higher nitrogen content, which is a reflection of the microbial characteristics of it. And that's going to then impart a lesser aromatic character and perhaps changes some of the competition.
So I'm quickly interjecting with a 3-minute warning.
Gotcha. So I'm going to wrap up here. So here's our overall trend. So I'm concluding. So overall, our properties of the PAC are most important. The mesoporous PAC performs best for toxins such as microcystins because of their size. The net charge is important because it influences interactions with these. And we've done some work with saxitoxin, which is positively charged, that further reinforces that. The organic matter competes, reduces sorption, but it does appear like there is a threat above which organic matter is not necessarily going to increase this competition. And in terms of recommendations or take home, if possible, determine which microcystin variants are present. And that's because the neutrally charged ones are removed most readily. And if that's the case, if that is what is present, then the type of the PAC becomes less of a factor, because I know the wood-based PAC is more expensive. If that is not possible, then a carbon that has a high mesoporosity is the best choice. And that was one, but other carbons that have high mesoporosity could serve just as well. And what should be noted is, again, reliance on the iodine number of the total surface area is not recommended, because they don't necessarily capture the actual porosity of the material. So more information is needed. Obviously, the contact time is important. So looking at removal on the basis of equilibrium that's done over a 3 or 5 day period is not recommended. And obviously, organic matter competes, reduces sorption. So if there's an opportunity, say through a pre-oxidation step, to deal with some of that organic matter, that will help PAC performance. Lastly, I'd like to acknowledge those who did the work. So Ashnika, Yenling, and Kate are prior students at Ohio State. Yujo, who's most of this work, this is her research, is still at Ohio State, and again, funding from OWDA and ODHE, and then the program, HABRI program, was managed by Ohio Sea Grant. Okay, so sorry if I went a little bit long. If there's time, I'd happy to answer questions.
I'll read one question quickly to you, John. Jennifer, I see your question. I will type an answer to that as best I can. But John, I'll read one question to you. And if you have a moment, do you mind typing some answers to the questions that have appeared in the Q&A box during the break?
Okay, I can do that, sure.
Sure, so I'll read the first one. Dr. Lenhart, this is from Liz Smith. Dr. Lenhart, you have identified ozone as ineffective for intracellular toxins, but ozone nanobubbles are being explored as a remediation technology for ambient water, thought to destroy first cells, then liberated toxins. Is treatment plant ozonation different somehow? If not, would ambient water ozone, if not, would ambient water's ozone use could result in problematic byproducts also, or are those particular to treatment plant processes?
Oh my, so there's a lot there. Try to unpack some of that. So it's my understanding that the issue with ozonation is kind of twofold. Ozone is ineffective for the extracellular toxins. It's good. Very good as a matter of fact, at degrading the extracellular toxins. But there's some literature that suggests that the resulting byproducts could potentially still be toxic. So it depends upon what they degrade into. And then it's my understanding that ozonation of cells is problematic because the disruption of the cell can then release the intercellular toxins, and then that can be problematic. So if the approach simultaneously can manage those released intercellular toxins, then perhaps it's a good approach. I guess I'm not familiar with the ozone nanobubble technology. I'll have to look into that. So I don't know if I addressed most of those questions or not.
The ozone nanobubble technology might get some address later by Dail's talk. I don't know. We'll find out the hard way, and it'll be fun. That said, we are flat out of time. So we are now going to enter a break. John, again, I will take Jennifer's question to the best of my ability. If you don't mind scrolling through the other questions and answering as you're able, that would be
Yep,
sincere.
I can do that.
So we will take a break. We will reconvene promptly at 1:55 Central. That's 2:55 Eastern, and you're in luck. I just checked and there's no line at the bathroom. So I will see you in 10 minutes.
All right. Well, thanks, everybody.
Welcome back, all. My Verizon tower tells me it is exactly 2:55, which means your Verizon tower might be telling you it's 1:55. So hopefully Will is ready. I'm going to go ahead and introduce Will. Are you here, Will?
I am here.
Fantastic. Dr. Osterholz is a research soil scientist with the USDA ARS Soil Drainage Research Unit in Columbus, Ohio. His research focuses on soil nitrogen, phosphorus, and carbon cycling in tile-drained agricultural landscapes. And Dr. Osterholz will address contributions of legacy soil phosphorus and phosphorus fertilizer to edge of field phosphorus losses. I'm going to stop sharing my screen so you can share yours, and thank you so much for presenting for us today.
Yeah, thank you for the invitation. I'm glad to be here. Let's see if I can get this started. Right screen. Hold on. There we go. All right, can you see that now?
You are seen.
Great. Okay. So
However,
Will, I'm Will Osterholz. I am with the USDA ARS Soil Drainage Research Unit in Columbus, and I'll be presenting some work today. I've been collaborating with Vinayak Shedekar and Kevin King to look at the contributions of legacy soil phosphorus as well as phosphorus fertilizer to edge of field P losses. So obviously this will be more focused on preventing phosphorus from getting into our water system. So I live and work in Ohio, so I like to always start with a nice picture of the beautiful green algae swirling through the lake. Unfortunately, the impacts on water quality are not as nice as the image. So in our group, we've been working a lot trying to address the sources of phosphorus that cause this problem. We know this has been a problem for the last 2 decades, really, in the lake. And lots of research has shown that phosphorus moving through the Maumee River into the lake is really the main culprit behind these algae blooms. So over the past decade, really, Kevin King with our research unit has developed this edge of field water quality monitoring network. So this consists of 40 paired fields that range in size from 1 to 18 hectares. So these are really field sized. They're located mostly in the northwest quadrant of the state. You can see on the map, the black dots indicate the locations of the sites. And that's the region of the state that drains into Lake Erie. It also is the part of the state that is heavily tile drained. So the landscape there is quite flat. The soils are pretty heavy. So subsurface drainage is really necessary for crop production in this region. And the management of the fields is really determined by the individual farmers who work the fields. And the treatments we apply to the paired fields include a whole suite of management practices, including tillage, fertilizer management, cover crops, crop rotation, gypsum application, and drainage water management. So at each of these fields, Kevin's team has instrumented the fields with both surface flume to measure surface runoff, as well as a V-notch weir to measure the tile drainage. So at both the flume and in the weir, the discharge is measured every 10 minutes, and there are automated water samplers to take samples for determining nitrogen and phosphorus concentrations multiple times a day. So I wanted to share some just general findings from this network before I get into the specific project I've been working on. So the left panel here is showing the annual DRP, which is, if you're not familiar, it's a dissolved reactive phosphorus. So that's the form of phosphorus that's primarily responsible for the algae blooms. And it's what we're mostly focused on in our research, although we do also look at total phosphorus. But today I'll mostly be talking about the DRP component. So each of these bars represents an individual field, and the red dashed lines represent the loading targets that have been set by the Lake Erie Task Force several years ago. So that's sort of the goal, is to be below that red dashed line. So what we can see is that there's a lot of variability between our fields within a year. And there's also quite a large amount of variability between years. So it's important for us to have a wide suite of fields that we can look at, and also a long period of record, because there is a lot of variability inherent in these measurements. But if we summarize this whole period, all of these fields across this whole period, that's what the pie charts are showing. And the orange portion of those charts is showing fields that exceeded those loading targets. So we can see that both at an annual time scale as well as the springtime period, the majority of our fields are exceeding the P loading targets. We've also seen that water movement in these fields is primarily through the tiles. About 80% of discharge moves through the tile drains, and the other 20% would be surface runoff. The phosphorus concentrations do tend to be higher in the surface runoff than the tile drains. However, even with that difference in concentration, the majority of the loads do move through tile drains. So about 60% on average of the phosphorus coming off of these fields is moving through the tile drains. And finally, we have also noticed that the P concentrations tend to increase with higher discharge conditions. So that just really highlights that it's these large flow events, these storm events, that are problematic, both in terms of the concentrations as well as the loads. Okay, so we know that there's excessive P moving off of these fields, but we can ask ourselves, what is the source of that phosphorus? And there's sort of two categories we can consider. And they're what I like to call new P and old P. It's sort of a simple terminology I like to use. New P is just what it sounds like. It's phosphorus that was recently added to the fields as fertilizer or manure. It's not yet become fully incorporated into the general soil phosphorus pool. Old P, on the other hand, is that background pool of soil phosphorus that's been built up over the period of years and decades. And we should care about trying to distinguish between these pools, because the mitigation strategies that we would use to try to address these sources are going to differ. So if new P is an important source, then we can think about applying fertilizer management approaches, things like subsurface phosphorus placement. But if old phosphorus is a large contributor, then we need to think about either trying to reduce the general pool of soil phosphorus or trying to intercept the phosphorus at the edge of the field. So how can we distinguish between these two sources using the edge of field data? So my approach has been to assume a risk period for new P loss of 3 months following each phosphorus application. So in each of these 3-month periods following an application, I predict what we would expect the old P loss to be during that period in the absence of an application. And so I do that using the discharge that we measured from that particular field. And then after I have that expected prediction of old P, I basically subtract that prediction from the observed loss. And then the difference between that observation and what we predicted would be considered derived from the new P source. So it's actually a pretty simple approach. I have it represented in an equation here. So new P loss is just the difference between the observed and the expected old P loss. And then old P loss is, what we expected the old P loss to be during that period, but it also includes all of the P loss that didn't occur during one of those 3-month risk periods following an application. So I have a quick example here just to illustrate this. So this is showing 1 year of P loss. This is DRP, and you can see there's a big spike in the P loss after an application in June. So in order to do the calculation, we come up with a prediction of the old P loss during that 3-month period following the application. And then we just subtract the observed or the predicted P loss from that observed P loss. So if we add some numbers to this example, we saw that the observed total DRP loss for this period was 0.27 kilograms. So the old DRP for this period was 0.08, and then the new DRP would then be the 0.19. So basically I did this calculation technique for our full data set. Again, it's 40 fields. We use data through water year 2019. And for our data set, we had 102 different applications occur across these 40 fields. 64 of them were chemical phosphorus fertilizer, and then 38 were manure applications. So let's get into some results. This graph is showing the annual DRP loads, both from surface as well as tile, for each of our fields. So each bar here represents an individual field, and I have the DRP load split into source. Oh geez. So the blue portion of the bar is the old DRP, and then the orange portion of the bar would be from the new DRP source. So what we can see is that across these fields, a lot of variability, like we saw earlier. Some fields tend to have a lot of DRP loss and others do not. We've noticed in the past that that's highly correlated with soil test phosphorus. So that's one of the primary drivers of this variability. And then if we look at the proportion of DRP from the two sources, we can see that for most of the fields, the new DRP makes up a pretty small proportion of the DRP loss. However, there are a few fields where it's a pretty significant portion. But the bottom line here is that on average across all of our fields, we could attribute 17% of our total DRP loss to a new P source. So that's P fertilizer or manure. So then the flip side of that would be the other 83% was attributed to old P or legacy soil P, if that's the term you prefer. Okay, so that's sort of the big picture. We can also dive into these individual P applications to see what they can tell us. So here is the 102 different P applications that we had in our record, divided into either chemical fertilizer or manure. In this graph, I also have split out the pathway of loss. So the surface loss, surface runoff loss is sort of the hatched portion of the bar. And then the tile loss is the solid portion. So again, what we can see here is for a lot of these P applications, the majority, there's very little new DRP loss. In many cases, it's basically zero. However, there are a few applications where the applications seem to lead to a large loss of that recently applied phosphorus. And so those are the ones that we should really be worried about. So what we can do is try to categorize each of those individual applications by the form of fertilizer as well as the application method. So that's what I've done here. Basically aggregated all of those into different categories. So we have the chemical fertilizer applications are the 3 bars on the left, and then the manure make up the rest of the bars. And then I've also split out whether that fertilizer was a solid or liquid form, and then whether it was broadcast and then incorporated immediately after application, or whether it was injected. So, you know, the very obvious insight here is that this yellow bar really stands out. And this bar is representing liquid manure applications that were broadcast on the soil surface. So we see that there's a much higher risk of new P loss when that is the application technique. And it's sort of interesting that, you know, again, I have it split out by the pathway where the hatched portion is surface and the solid portion is tile, that for these broadcast liquid manure applications, it's really the tile discharge that's contributing a vast majority of this P loss. If we look at the surface runoff, it's really not any different from any of the other application categories, but the tile discharge really tends to bring a lot of DRP off the field. So in our area, our soil textures are quite fine, and we tend to get a lot of macropore formation, whether it's cracking in the soil or earthworm burrows, things like that. And so the hypothesis is that when this liquid manure is applied on the soil surface and we get a rainfall, basically it carries that newly applied phosphorus straight through those macropores down to the tile drains where it can leave the field. So the last graph I have here is looking at the application timing. So I've narrowed into just the broadcast applications. So this is either broadcast fertilizer or broadcast manure. And I split that out by which month of the year they were applied. So we can see that when these broadcast manure applications occurred in December, we tended to get greater DRP losses. And then the flip side of that is, you know, April, we saw very few new P losses when that was the month of application, as well as September and October. So those seem to be times of year where there was less risk of this new P loss. So a summary here of some of our initial findings from this analysis. The big picture here is that the new P source accounted for just 17% of our overall DRP losses. And I didn't show any of the total phosphorus results, but for total phosphorus, it was an even smaller proportion. It was 11% of the total phosphorus loss. So then the remaining losses could be attributed to the old P, I should say source. I'm not sure what happened there. And then we saw that the riskiest applications tended to be those liquid manure applications that were surface broadcast. And again, that was DRP lost through the tile drains accounted for the vast majority of that. And then we also saw that broadcast applications in the winter, so that December time period, had the highest risk of new P loss. And then early fall had a low risk. So that sort of agrees with some of the best management practices extension would make. You know, that winter application period does seem to be the problem in our data. So a few implications here of these results. You know, for our region, it seems like the phosphorus fertilizer management, so things like 4R, is going to have a limited impact on P losses. And I've specified here that this is a short-term picture. You know, obviously, if we apply less phosphorus fertilizer over the long term, that's going to help address both the new P losses, but it'll also help address those old P losses that made up the vast majority of P losses for our data. However, on the short-term timescale, we're only really messing around with about 20% of the loss when we try to fiddle with the fertilizer management. Of course, that 20% can still be important, but my point here is we really need to place some additional focus on trying to manage old P losses, because, you know, those are the vast majority of the problem. So some possibilities we could look at, you know, the obvious one is drawing down soil P. So that's going to take a long time, but it's something we need to try to do. Keep our soil test phosphorus on the low end of recommendations if we can. We can try to manage hydrology. We know that, again, I mentioned earlier that our concentrations of phosphorus in the runoff tend to increase for the high flow events. So if we can try to buffer our high flow events a little bit, that could help address these old P losses. We can try to manage phosphorus stratification. We know that there's a lot of fields in Ohio that have a high degree of soil phosphorus stratification. So if we can help move some of that phosphorus deeper into the soil, whether that's by some targeted tillage or potentially injecting our phosphorus fertilizer to try to move it deeper into the profile, that's a possibility. And then finally, we can try to capture phosphorus at the edge of the fields or downstream, again, using things like P filters or wetlands or things of that nature. Okay, so if you're interested in learning more about some of the findings from our Edge of Field network, we have a pretty good list of recent publications that have come out. So I would encourage you to look some of those up in your spare time. And then I'd like to acknowledge, of course, the cooperating farmers are vital to making this Edge of Field work happen. And the team at the Soil Drainage Research Unit has been amazing. You know, 40 fields is no joke. And a lot of them are 2 hours from Columbus. So they do a lot of really, really great work to make all this happen. And then, of course, the funding sources. We have a large list of organizations that have contributed to this work. And we're very grateful that they've made it a priority to help us generate some data to help back up some of the ideas that people have been proposing to try to address this problem. And with that, I think I have some time for questions. And if there are any, I'd be glad to try to answer them. Otherwise,
you do indeed, Will, and we have some good questions as well. So thanks so much.
Happy for emails as well.
Sorry, there's a bit of a delay, and so we end up talking over each other thanks to Zoom delay. But thanks again for your presentation. It was excellent. First off, from Coker032, which I'm only guessing probably does not appear on a birth certificate, but Coker032 asks, "Hi, Will, how do you measure soil test phosphorus?"
Soil test phosphorus. I didn't talk a lot about that in this talk, but we do measure soil test phosphorus in all of our fields. We do it on average about every 3 years is sort of our sampling schedule. And we try to do a sort of typical agronomic approach where we try to emulate what, you know, commercial farmers would be doing. So we go out and take, you know, grid sampling or at least zone sampling on all of our fields. and have them analyzed by a commercial lab for usually Malic-3 extraction. We've done some work looking at different fractions of soil phosphorus besides the Malic-3 extractable, but you know, that's the standard, so that's what we tend to fall back on. Hope that answers your question.
And I assume that you don't mind taking email questions if somebody wants more detail.
Yeah, of course. Yep.
Thanks. So Sandy Bin asks, which of the legacy fields for old phosphorus have fields that apply manure versus fields that use commercial fertilizer? How is the P application rate for fertilizer factored into this process? Are any of the edge of field tests located in the Auglaize or St. Mary's watersheds where manure application is occurring?
Yeah, so we have a mix. This is mostly about the history of our edge of field sites, I think. And we have a mix of sites that some have a pretty strong history of phosphorus application through manure primarily, and those do tend to be our fields that have higher soil test phosphorus. You can see we have a little cluster. I don't know if you can see this, but there's a cluster down here. That's right around the Grand Lake St. Mary's area. So yes, we do have some representation of that region and sort of their sort of unique situation. They do tend to have much higher soil test phosphorus levels than a lot of the rest of the northwest portion of the state. So yes, we do have some fields with high legacy, you know, this old soil phosphorus, and those do tend to be the fields that have gotten P application and still do get P application through manure. Yep.
Great, thanks. James Spetz asks, oops, hold on. It's scrolling around as people move questions. And James Spetz asks, can farmers maintain yields in most situations without new applications of phosphorus in order to draw from that existing old phosphorus? How long do we estimate it may take to draw that old P down to acceptable levels on average? Good luck with this one.
Yeah, that's a tough question. I'm not sure I'm equipped to fully answer that, but there has been some work by some people at Ohio State. Steve Coleman comes to mind, who have looked at, you know, the importance of P fertilizer for maintaining yields. And a lot of that work shows very limited responses to P fertilizer in Ohio. That's not to say that there aren't a lot of farmers that do think they get fertilizer response. So it's hard to refute that fully, but I think there's definitely opportunity to at least cut back on fertilizer rates. A lot of farmers, I think, are very reluctant to move away fully from P fertilizer. They really like starter fertilizer. So I think it's going to be a tough slog to get them off of starter fertilizer, but I do think there might be some room to reduce the rates of these sort of basal applications that tend to happen in the fall. So that's where I think, you know, a lot of the potential lies. In terms of the rate of drawdown, that's a tough question. It varies a lot by soil type. Heavier soils like we get in the northwest portion of Ohio, they tend to draw down more slowly. They're more highly buffered in terms of the phosphorus. You know, to give you specific numbers, you know, it's, if you want to draw it down 10 or 20 parts per million, if that's sort of your target, that's going to take you probably a decade to get that degree of reduction. So it's a slow process, but it's possible, and I think it needs to happen. So the longer we delay, the longer it's going to take to reach the goals. So hopefully we can continue to make progress on that front.
And even if you could calculate a meaningful average, I imagine the standard deviations around that average would be really wide.
Yeah, they definitely would. again, because it just varies a lot by soil type, and it's tough to really narrow in on an exact number.
Okay, thanks.
The time scale we're talking about.
Thank you. Lynn Klontz asks, how do you engage farmers to cooperate?
That's a great question. That'd be better asked of Kevin, because he's really been the point person on developing these sites and getting this network up and running. I know a lot of it is just word of mouth and personal relationships. So we have good relationships with the farm groups. And so a lot of our cooperators actually are highly involved with, for example, the Farm Bureau or the corn growers. And they tend to be the ones that show interest in helping us try to answer some of our questions. We also get some cooperators coming through extension, NRCS folks, you know, that they'll have contacts, farmers on the ground who show some interest, and they connect us. So it's a lot of that sort of organic relationship building.
Great. I think that's all that we're going to take live. There are still a handful of questions remaining. Will, do you have some minutes to type some answers?
Sure. Yeah, I'll stick around and see what I can do in the.
Great. Thanks so much for an excellent and really relevant presentation.
Yep. Thank you.
And finally, to end the afternoon session, I'd like to introduce Dr. Dail Laughinghouse, a broadly trained phycologist working with both basic and applied algal research from tropical to polar regions. His background includes systematics, ecology, harmful algal blooms, cyanotoxins, phycoremediation, biofuels, metagenomics, and metatranscriptomics of marine, freshwater, and terrestrial environments. And I'm going to congratulate myself for having read that. Some of the current research in the Laughinghouse lab focuses on diversity, ecology, and toxicity of cyanobacteria, environmental influences on macroalgae and microbial eukaryotes, protists in parentheses, novel applied uses for algae, bioremediation, and the detection and effects of bioactive compounds. Research on testing different algal treatment methods with industry and other stakeholders is ongoing in the lab. The Laughinghouse lab is always welcome to collaborative research. Coincidentally, some of which I am actually currently about to engage in. So thank you, Dail. I have stopped my screen share so you can share yours. I'm really looking forward to your presentation. Thank you for joining us today.
Thank you. Can you hear me?
I can.
Okay, I'm going to share my screen. And while I share my screen, I just want to make a comment. Thank you for everyone being here, especially if people have looked at the news. I can't get off my phone. So half of my thoughts are on the CAPA being breached in Georgia, and now other part on the HABs. So anyways, I'll do my best to focus on HABs right now, okay? But anyways, let me see my screen. Let me just make it big for myself so I can actually see what I'm doing. You know, it's not like we haven't done Zoom for a long time and I can't figure this out all the time. There we go. Right, good. Okay, so I will talk a little about chemical treatment, but I'm going to talk a little bit overall first. Again, thank you for inviting me. My name is Dail Laughinghouse. So I'll talk about mitigation and treatment of cyanohabs. I'll also make a comment about some other blooms that exist, like benthic, terrestrial, mats, and all that, so that we have a larger idea of what I think about when I think about treatment or mitigation. I think about how to do the marine ones, how to do the terrestrial ones in the greenhouse, how to do the aerosolized toxins, how to do aquatic ones, how do you do pelagic, how do you do benthic, and all changes, right? So here, so have an outline, like I just gave you a general overview of what blooms are, proliferation, crusts, and mats. I'm not going to go into this. I'm sure this morning that was there that much. Then we're going to go to proactive management methods, then reactive management methods, then managing blooms directly and indirectly, and then managing toxins, all in 30 minutes, yay. So if you guys want to go watch the news, I'll just give you the conclusions already. So we need more science-driven data on current and new treatment methods. That's easy. Also, we need to see or assess the feasibility for different systems at scale up and cost. Remember, it's no size fits all. And people tend to black box this, and you shouldn't black box species, you shouldn't black box treatment methods, you shouldn't black box waters. Again, like I said, not all waters are the same, not all cyano and algae are the same. Efficacy of treatment methods differ. And we need long-term data on effects of chemical formulations that are, novel ones and those that are still EPA registered, proposed bacteria and their byproducts, proposed enzymes on the environment and on non-target organisms. So I always like to begin my talks with this here, this slide saying algae and attitude. So even though I fell in love with algae about 18, 19 years ago, I can't remember when I started, when I talk about it, still people are like, what are algae? But anyways, that we all know that. But I think it's funny because, I do work with scum, and I mean by pond scum, not necessarily colleagues. But so it's pond scum, frog spittle, seaweed, green slime, pea soup, guacamole, ground boogers. These are just wonderful names we have for these organisms, or this organism, if we talk about cyano, that gave us the oxygen we breathe. So I always like to say, if you guys don't care about cyano or algae in general, why don't you hold your breath right now? Which many of us probably are doing because of the news. But anyways, just to show some pictures, that's me on a side canal in Okeechobee, and then that view on the right here, I'm not sure if you can see my cursor, is the view from my apartment. And it is a bloom all the time. I live in Fort Lauderdale, and it is right now 73 degrees. This morning was very cold. It was 55 degrees Fahrenheit, and now it's 73 Fahrenheit. This is a bloom all the time. Talking about that bloom, the people that have heard me talk about it, we've used that bloom for testing. We've isolated species from that bloom. I think there might be some new species from that bloom as well, or that water. And I know that I talked to my HOA one time and said, asked them what they were doing to the water, and what were they sticking as fertilizers and everything. And they were asking me questions to mitigate. I go, no, please don't. If you see some people, in the water or sampling, just make sure, it's fine. I just don't stop it, because that's my test pond. So again, these you've seen, but just to get a thought moving from, because before we were talking about drinking water, and then we're talking about fertilizer. Let's get our thought back into scum. So there are numerical values for planktonic blooms, but I'm not going to get into that. That was probably this morning. But anyways, planktonic blooms are dense growth of algae. We know very well they produce several compounds, and many being toxic. Another interesting thing is that they're able to harbor pathogenic bacteria, so on their mucilage. So even if those may not producing any compound or toxin, they may have, well, they're always producing some kind of compound, but they may be producing pathogenic bacterium that's maybe worse than what toxins do. And again, contain taste and odor compounds and have been observed for centuries. I think 1188 was one of the first observations that have been, I saw that somewhere, but back in the Mayan era, the Aztec era, they were looking at blooms. And there are the data saying that a lot of, there were people that were sick by blooms due to that. Again, not all blooms are the same. I get all these questions because I also have part of my FTE is extension. They send me pictures like this and ask me, what is a species? And for any of you guys that know, I have to explain to people that these are microscopic. But anyways, that first on the top left with the turtle, that one was more like an Aphanizomenon or cylindro bloom or Raphidiopsis bloom. So Aphanizomenon, cylindro, which is now Raphidiopsis, that was in there. And actually, what's interesting is that pond up here with the turtle, it was around Hialeah, in larger Miami. There was actually an aerator or aeration system in the thing. As you see, it did not do a very good job. Down, on the bottom with two of the people that work in my lab, David and Forrest are sampling, and the Army Corps person is hiding. I have to say thank you for the Corps who's here. You guys have helped us a lot with transport in the field. I'm very, very happy and have supported us a lot. I'm very happy with everything that you guys have done for us, and thank you so much for the Corps. So we're sampling there. That was, and then on the top right, that's also from Lake Okeechobee. And if any of you guys in the audience were at Itcha in Nantes in France in 2018, this canal, or this kind of river canal system that was right beside the conference center, that had a Planktothrix bloom. So that's what was in there. Again, some pretty pictures. Just this, what I'm trying to show you guys is that, I guess you guys probably know as well. I don't know exactly everyone's background. We're 400 some people. Not all, we talk about cyano, we talk about algae, but they're different, right? Species matter in relation to evolution. Species matter because they evolved with some nutrient requirements, and they also evolved with certain compounds, and they also, treatment methods differ. The efficacy of treatment methods differ. And I'll show at the end that they differ even at the Microcystis aeruginosa to Microcystis weissenbergii that detailed system, that detail. So on top left here, we have Dolichospermum spiroides. On the right, Planktothrix isothrix. And down here on the bottom left, for those that are taxonomists, understand that when we see blooms, we see this. Those are 3 different species of microcystis in there. The clathrate one is aeruginosa, of course, and I think the other one is Botrytis and maybe Protocystis. I have to go back because these are difficult to see. Of course, and then you know towards the bottom right, this is Sphaerosomatsis torcus reginae, and these are all from except for the top 2 are from Brazil. The all bottom ones are here from our waters of Florida. What about other blooms? Okay, so you know when you're thinking about treatment and what makes it grow or how to mitigate them, you know, I know we're focusing on aquatic blooms, but also there are benthic blooms, right? These are also aquatic. They produce other, you know, this is a review paper that I was involved with several people that are probably in the audience, and we look at they are, you know, the causes are different, or different nutrient factors can lead to these benthic proliferations, and also the treatment methods are different, and a lot of these are in flowing water as well, and treating a flowing water is very different than treating a pond or a lake, right? So all these things have to be thought about when you have a treatment plan and part of your toolbox that you do. But anyways, that's, I just want to put that. Also, you have toxins produced in soil crust or microbial mats. This is just pictures I had from the Arctic. But anyways, just to have an idea that they're also there. Do we need to mitigate them or not? We kind of do, because we just published this paper, came out it's coming out not here, with a new species of Inningainema. If you look here on the right, Inningainema is very close to Satinema, and it looks very much like a Satinema. The growing in greenhouses with food production, and they produce nodularin. So nodularin is very similar to microcystis for everyone that knows, and also has biosynthetic potential for producing microcystin LR and adipinopeptide. So, and it actually has a quite large genome if you're looking here. But anyways, what I want to say here is, you know, registered products for greenhouses are different than registered products for the water as well. But you know, again, what is feeding that growth? How do we treat it? How do we remove it, right? And so this is something that I just want to keep in your mind, that it's not just water. And moving on, other things, we have other greenhouse nuisances, as you can see here. And this is the 3 new species that are being published from Florida, from the same area of ornamental plant production. We're looking at these actually aerosolize Hormogonia, which was interesting. If you see on the B here, those little pieces are little Hormogonia, and those were aerosolized, not necessarily toxic, well, even though there's some toxic compounds found in this genus. Organic decomposition through aerosolization is also a problem for airways. So this is a big problem for greenhouse workers. And again, so how do we treat that? How do we make sure? And if any of you guys have worked in greenhouses, especially in tropical, subtropical areas, these things form huge mats, and then they dry, and many people just kind of shovel them out, and you have all that dust. That dust is cyanoparticles, and other stuff as well. You know, a lot of pesticides and chemicals as well. So just wonderful stuff to be inhaling. And of course, we also have novel marine benthic cyanobacteria in Florida, and several are toxic. Though we're not drinking water from there, though how do these compounds affect the marine food web with coral bleaching and all corals? You see these benthic cyanomats going over and just covering things, and Val Paul down here in Florida does a lot of work with that and has shown that. And just, you know, just something that we think about, or I think about, what's causing that, but also how do you mitigate that? But anyways, let's go back to water, right? Since this is why you asked me to come. But hopefully I didn't confuse you guys too much, but just gave you a view into other things to think about, okay? So there's a bloom, and now what? Before I started working with treatment methods, you know, I never really thought about, okay, you know, we thought about causes of blooms. I thought about what they are, systematics, genetics, genomics, all the wonderful omic words, but I never thought about, okay, now what do we do? Okay, of course, there are both proactive and reactive approaches to cyanobacterial bloom prevention and treatment. But before, I just want to make one note here, and remember, any product or device that claims or implies to mitigate a pest must be registered under FIFRA by the US EPA. Okay, so that's just for the knowledge. So if any of you guys are working with any people that sell any compound, and they say it's an algicide and it's not registered, that's a breach. That's against the law, and there can be heavy fines. But anyways, so this is a small aquatics article that I worked with Dave and West on, and we looked at just I like this little table that we made, looking at proactive versus reactive, and of course they can be chemical methods on the physical, mechanical, biological. And so, for example, on the proactive methods, you have nutrient mitigation, water quality enhancers, and dyes. Then you have barriers as physical barriers, watershed alteration, stormwater control methods. On more the mechanical proactive, you have aeration methods, oxygenation, fountains, artificial mixing, withdrawal, dredging, harvesting, skimming. Again, these are just some. And again, proactive on the biological side, you can have biomanipulation of the food web, these magical bacteria, native plant establishment, floating islands, constructed wetlands, buffers, algal turf scrubbers, all these things that are just wonderful. And again, you see the toolbox. Every method or every proactive, reactive methods, everything I said, I believe that all these different methods have their place in certain areas or certain water bodies. Instead of saying one is better than the other, I would say that some work better in certain situations over the others. Another word I don't like to use is control of algae, because then people actually would control them. I like to use the word management, because then we can accept the fact that they're not going away, right? But let's go back to, I went on a tangent, I'm sorry. So let's go back to the chemical reactive methods. We have registered algicides, which I'll get into, dyes, flocculants, polymers, coagulants. On the physical reactive, we have raking, skimming, shade cloth balls, flushing. On the mechanical side, you have sonication, ultraviolet light, cavitation, ozonation, which was spoken about with Dr. Lenhart before. And yes, Eugene, we are working, like Eugene said, I am collaborating with them and with him and others of the OSU in a new project that will be 3 years. One part is looking at that. And I'm very excited about that. I know Heather as well. I know you're on here, so thank you so much for inviting me for that. Then on the biological side, reactive, we have plant extracts, enzymes, microbes, at fish, invertebrates, many things. Okay, so again, if you guys, my email's on here afterwards. I do speak fast, I know, and again, I have to stop looking. My phone keeps on popping up with messages, and I hear it. So because all friends are just sending me things, but I am happy to send you any paper that I cite here or have a conversation. But let's continue with the toolbox. So again, we all understand that long-term nutrient mitigation management is needed, right? But if you tell a homeowner that has a $4 million house on a lake that they'll have to wait 200 years because the legacy phosphorus won't go away that easy, that's not something you tell. Anyone that works with extension knows that that's not what you tell the people. They don't want to hear that. You have to explain that, but you also need to, but you have to explain what the long-term needs are. However, what can we do in the short term? But also understand what expectations you would have from those short-term methods, and also the potential secondary effects of any chemical compound you put in the water, right? But going back to proactive management, I've explained these already. Reducing nutrients, aeration, temperature, climate change, we do our stuff, we do our part. Bioremediation and dyes. This is already explained, so I'm not going to go into it again. There are some other ones that didn't explain on the chemical part of nutrient remediation. Remember, some of these I've heard sold, like alum, polymers, iron, calcium, all these chemicals I've heard people try to do the sell as an algicide. In the United States, they're not an algicide. In other countries, they are used as algicides, but in the US, no, they're not. I'll go into Foslock, which is the LMD, a little bit more in some of the slides. So of course, this is what we know. Reducing nutrients is important, right? Especially nitrogen and phosphorus. And thank you, Will, for speaking about the different phosphoruses, because I put P as P and N as N. But if any biogeochemists are there, they're probably getting so mad that I'm not speciating these out like I do when people talk about cyanobacteria or chlorophyll in general. So I just, I do acknowledge that there are different forms of Ns, there are different forms of Ps, and certain ones are better for certain species over the others, but that wasn't the focus of this talk. Again, if you read the literature between phosphorus and nitrogen, it's so interesting because you have this significant different opinions by different groups. But I do like the image on the bottom. That was one from Hans's work, where it takes 2 to tango, looking at both N and P. So of course, we talked about, Will talked about phosphorus, so I'm not going to go into that, but cyano are phosphorus limited, and phosphorus is the limiting nutrient in fresh waters. Okay, but which nutrients deteriorate water quality and control algal blooms? Now that answer depends on the water body. Understand that different nutrients drive different algal groups, but not just different algal groups. Within the cyanobacteria, different nutrients will lead to different species of cyanobacteria blooming or growing. I find this is fascinating. And then I'm not even talking about the microbiome in there, which I'll make a little comment about in a second. So just to prove that what I'm talking about, this is some work we did in Chile. If you look on this towards the colorful, I'm not sure if you guys can see my cursor, but anyways, you can see that with land use change, so these are more tree plantations, urban area, and tree farming, we see an increase in nutrients and we see an increase of cyano, right? And then on the bottom where there's more forest land and not as much manipulated and not as much increase in nutrients, we see less cyanobacteria and mostly the other cryptophytes, ochrophytes, and the different in diatoms, the flagellates and all those fun little ones that move around. Actually beautiful as well. And there's some of them swim, like ballerinas. But nothing swims as unique as Carenia. That's an interesting one to see swimming. But so continuing this, what about herbicides? So we're not the first group to see this and do this work, but we're working on it right now. So herbicides as a nutrient source. So if you look on the right here, this glyphosate is, glyphosate has been shown to increase cyanobacterial growth. And many people here, and if you look at the graph here, that yellow line is a very low concentration of rhodio, which is the aquatic glyphosate. So most of the time, people, when we say glyphosate, people think about Roundup. Roundup is terrestrial, but there are aquatic forms. And so if you look at the glyphosate breakdown between the pathway A and B, if you keep on going around, you see AMPA, you see Ps, you see this very nice, the ammonium here, but then you also see sarcosine and glycine. All these here are nutrients. And so what we noticed is that when there is phosphorus in the water or phosphorus in the medium, and we add a very low concentration of especially of rhodio in this case, but also glyphosate, there's an increase of growth, which is significantly higher than the control. So again, Dariusz Giga has done this work also in Poland, and some other groups have looked at this. But we are interested in understanding more of what is going on. What is going on with the breakdown products? What is actually causing this growth? Due to the usage of glyphosate worldwide. Again, just to add more complexity when we're talking about nutrients, remember that first, the blooms have several species of cyano and other microalgae. However, there's this consortium of many bacteria in there, of fungi and viruses and everything. But a lot of these different pathways, which is the cyanobacteria may not have, are in the bacterial, the metagenome, right? So let's say we decrease nitrogen, and we can say, okay, we can decrease nitrogen because microcysts can't fix it. How or whatnot. Look on the nitrogen cycling here. The metagenome on the microbiome has the nitrogen cycling capability. So again, it's kind of like when we're trying to look at nutrient mitigation, we tend to simplify, and that's the way it happens. To put models need simplification, right? We can't put all these parameters in there, but it's just so much more complex, and that's why we don't have an answer yet. But and I don't mean to confuse people because I barely understand this. This looks like an electric circuit here on the right. But what I can say is that you see pathways that are in the microcystis, in this case. You see pathways that are on the epibionts, and you see pathways that are detected in both. Right, so this is fascinating, and to understand like how a treatment method, how it affects everything, I think is something that's very interesting. But now going back to something else we know, so aeration. So aeration is another preventive way of doing this. Why? Because it's to prevent scumming and to prevent stratification of the water, and it also is used to oxidate the benthic layers to decrease internal phosphorus cycling. And that's the legacy P comment that was talked before. So in this little graph here, you can just see over time of season from March to October. No mixing, you see a huge cyanobacterial bloom. And then with mixing, you see diatoms. And if you go over here from Petra's work over here, turbulent diffusion up in the top right, you see that diatoms and green algae win with more turbulence, okay? However, what happens with P resuspension when you're doing this? What happens when you turn the aerator off? The design is very important, and there's several companies that have worked in this research done, both anecdotal and peer-reviewed research looking at how to install these aerators to make them work or to make them efficient. Continuing here, blue dyes. So dyes are another way. Golf courses like them because, well, golf courses usually have the dye in copper. But anyways, you know, they like them because it's non-toxic, right? It's like a food coloring, right? And it's registered for use in logic systems. And it's like a physical control. So this blue dye absorbs the wavelengths of the photosystem 2 with between 550, 650. Where's 550, 650? Look here, down here on the wavelengths. That's between phycocyanin and phycoerythrin, right? And phycoerythrin is used by estuarine, well, marine cyanobacteria, and phycocyanin is used by the freshwater cyano. So it's like this, they're competing for light. And if you look at this old, this graph here, you see that it's the white bars control. Well, I love these different kind of vertical. Why? Nothing inside the bars control. These lines, these vertical lines are 1 ppm. Then you have 3 ppm and then 5 ppm. And you're seeing that with increased glyphosate within the legal rate, you do see decrease in the growth of these different species. The beginning here is Aphanisamina flozacco cyano. This is Dolicho now. And then you have the greens here. And I think this one would be a diatom. So let's go continue. And then we did this work. You can see us down here in the field. This is winter Florida. So, you know, the clothes that we wear is a bit different than people where you guys are in the Midwest. But anyways, and here we're looking at the effect in situ and liminal corrals of aqua shade on the phytoplankton community. So on the left, November 7th, if you look at the bar, it says control 0 meters and control 1 meter, because we did sampling at the surface and at 1 meter to see if there's a difference. And yes, this color, this kind of blue-green color are the cyano. And you see that at both surface and at 1 meter, that using the dye, there was a decrease in cyanos. Now in January, we really didn't have that many cyanos, but we did the same test again. It was an ongoing test for months. We noticed that it still affected the community structure in a different way, right? And so that that's interesting. And so yeah, here's some work. Yes,
it's a 4-minute warning.
Okay, I'll talk faster. I'll go through. So here's some work by just by CPRO that they shared with me, looking at Foslock and their application. So Foslock, remember, it's not an algaecide, it's used for phosphorus control, and that use was able to, you know, in this case, it caused less or decreased the chlorine from the lake. We've done the same work. What we've noticed about this is that that if you use, but our idea was, can Foslock sediment microcystin, right? So we noticed that high concentration of Foslock and high concentration of microcystin, yes, it was able to sediment it, and it worked. This is more of a reactive method, the one I did before, but I won't talk about it because John talked about it a little bit, using chars or biochar. Here's some work with biochar, and we've done some work looking at different feedstocks like sargassum, and can it be a use of biochar? The paper's there, and these are just interactions between the hydrophobic and hydrophilic. Hydrophobic hydrogen bonding, just the different ways that these the microcystin LR is able to attach or enter these different compounds, these different biochars. So for finishing up is reactive. So I've already talked about these, the different biological methods that exist. Again, this is part of the toolbox. You can use natural compounds. Here's a paper that uses natural compounds, and they have cyanobacteria up here, chlorophytes, and diatoms. And as you see, with more with the aqueous extract with higher concentrations, they do have a decrease in cyanobacteria and other algae. So it is a method, but how to make it large scale? Can you stick it over Lake Okeechobee? That's another question. I love this slide here that I went to talk by Melkonian. These little algivorous, these vampirillid amoebae. Of course, this is kind of, I'm making like a spatial what do you call it? Just not saying this in a work, but I just think it's an interesting idea of using these amoebae to eat all the algae. Now we have what I'll finish with our US EPA registered algicides. So remember what I told you guys before. So we have diquat, endothol, flumioxazine, peroxides, 2 peroxides, sodium granular, which is sodium carbonate peroxyhydrate, liquid, which is hydrogen peroxide, peroxytic acid, and then 3 coppers. Some are chelated, some are acidified, some are regular copper sulfate, and then we have adjuvants that go into this to make them more efficient. So copper disrupts electron transport, it prevents cell division, it inhibits enzyme catalase, inhibits protein, interferes with cell permeability, and with all that there, I'm not going to say it, but one thing that's important to note, not all waters are the same. pH, alkalinity, temperature, hardness, etc. affect the efficacy of copper in waters. And here's an example of using copper sulfate and on the top and copper and cutrines for chelated copper on the bottom for Plankton agardii. And what Alyssa saw was that the chelated coppers, and this has been shown before by other people as well, and is what's known as chelated coppers are able to work to control target cyano at lower concentrations, just copper sulfate, because there's a move away from copper sulfate, but it's copper still used, but other forms of copper. In this paper here, we're looking at formulations of copper ethylamine complex. So if you look at Captain XTR and Cutrine, this is Lembia now. Now we're talking about benthic. You're noticing that the Captain XTR is the black circles and the white open circles is Cutrine. So 2 different formulations, 2 different products with the same active ingredient that you see that they work differently. So not all products are the same, but they worked better than copper sulfate. That's what I'm trying to say. The chelate, in this case, they work better than copper sulfate. Now peroxides I'm gonna end with peroxides. Those are what people consider more benign to the environment in this country, but if you talk to people in other countries, that's a terribly oxidizing agent. But anyways, we look at it as more benign because it breaks down quickly into oxygen and water, right? It's more effective at high light intensities and it's less effective at low nitrogen and phosphorus concentrations. And some of this work here, BioSafe shared this with me, that after their peroxide treatment, there was a huge decrease in cyano. This here is one of the traditional papers by Hans Matthias and Jeff Huizen, looking at a lake in Groningen, outside Groningen. And in this here, they noticed that after peroxide treatment, there was a huge decrease of cyano, but not as much as zooplankton at the relatively low concentrations. And of course, after a few days, the peroxide was no longer in the water. And in this case, they found that within a few hours of treatment, more than 70% decrease in photosynthetic vitality. And then there was 99% collapse in cyano and microcyst concentration within a few days. So I have 4 more slides to end. So in this case, we did work looking at Florida blooms and looking at 15 products. And what I can tell you is that they don't all act the same. So this one is on Microcystin weissenbergii, which is not aeruginosa. The next one I'll show you is aeruginosa. And actually, no product actually killed weissenbergii all the way. Weissenbergii, if you look over here at B, they have this interesting mucilage. And what's interesting for those that are taxonomists or have seen cyano is that when you put these algicides in there, they even increase more mucilage. It's so interesting to look at, I think. And we've done some work as well looking at dyes to see the penetration of these compounds inside to see the efficacy in that way. But anyways, what I want to show here, and I can send you this if someone wants to, is that products act differently. And if you go to this paper that was just published looking at 11.1 on aeruginosa and toxins, you actually see that with different compounds, you actually got to almost 0, with some formulations. We didn't see that with weissenbergii, okay? And also, you see at total microcystin concentrations after 1, 4, 9 days, that with these different compounds, you do see that you can go to 0 or almost nothing of microcystin in the water as well. So this here's just to show visually what efficacious treatments look like, and that's one was copper sulfate and one is hydroperoxide, and this is to show you what looks like when it's not working, right? And what's interesting is that this is a peroxide here on the right that worked, but the granulated peroxide did not work for this species, which is aeruginosa, but the granulated peroxide worked very well for the Microcystis weissenbergii. So that's interesting, that I found very interesting, and some people don't think microcystis have a species. So anyways, now I am finished, and here are is my contact information, and I'm sorry for going over, and feel free to contact me at any time.
Well, thank you for an excellent and diverse talk, Dail. We don't have time to address questions live, but do you mind fielding some questions by typing to those for at least a few minutes after we wrap today, before we release those who are signed up for our discussion to follow? Do you have a few minutes to type the answers, Dail?
Yeah, I'll go. I see the Q&A right now. There are 4 questions, right?
Thank you so much.
Yeah.
So watch for those replies if you've asked for them, folks. And with that, I'm going to close today's presentation sessions. If you have registered in advance, we will have a facilitated discussion to follow, beginning in theory at 3 o'clock Central, 4 o'clock Eastern. We'll probably be taking a few minutes at least to shuffle folks in before we get to the full session. Before we do move to the full session, I want to thank you all for attending, especially given with the strange news that is evolving as we speak. But I also want to thank our sponsors, Utrofix, and especially FlowCam, and I can't read the whole because of, and Limnotech, of course. Thank you very much. If you have registered for the facilitated discussion, refer to the email where you referred with that registration information, and I will talk to you there soon. Thank you again. And I also look forward, we will resume again, of course, tomorrow morning at 9 a.m. Central or 10 a.m. Eastern. And we'll have another fine program for you then. So thank you again for attending this first of our Harmful Algal Bloom Symposia. And I really appreciate your attendance.